Hello everyone, my name is Devamitra Chakraborty and I'm a PhD student in the Material Science Graduate Program at University of Rochester, and today I'm presenting our work on selecting optimal substrate mounts in THz time domain spectroscopic imaging of murine radiation treated pancreatic ductal adenocarcinoma, or PDAC in short. This work has been done in collaboration with Gerber Lab at the Department of Surgery at the University of Rochester Medical Center. So basically, in this work, we report a method of selecting optimal mounting substrate and tissue thickness for performing THz time domain spectroscopy, or THz TDS experiments in transmission geometry. Here we are trying to characterize the difference between the stereotactic body radiation therapy, or SBRT treated and untreated PDAC samples. We found out that 50 micrometer thick tissue samples, when they are mounted on cellulose acetate films, offer the largest contrast between the treated and untreated groups. And we attribute this difference to the presence of collagen deposits and DNA fragments in the SBRT treated tissues. So to present a brief background on terahertz time domain spectroscopy. So terahertz is this wave which locates in between the microwave and the infrared region at the electromagnetic spectra. As you can see that the wavelength is in between 3 millimeter to 30 micrometer, which means that it has a larger wavelength compared to the optical wavelength, which makes the scattering much lower into the biological tissues. Also, this is known as the molecular fingerprint region of the electromagnetic spectra because lots of low frequency motion such as molecular vibration or molecular rotation falls into this terahertz region. Terahertz is transparent to the materials consisting of nonpolar molecules. Terahertz is also strongly absorbed by water. That's why we can sense the difference between the smallest changes in water between two different type of tissues. Also, this provides micrometer spatial resolution and femtosecond time resolution. Also, since this possesses much lower energy, terahertz is non-invasive and non-ionizing. So that is why terahertz time domain spectroscopy can be used for biomedical imaging. Now I'm going to present a brief background about pancreatic ductal adenocarcinoma, or PDAC in short. So PDAC is the third most common cause of cancer deaths in the United States, and most of the patients are diagnosed with an advanced disease where there is no curative treatments. Although SBRT-based approaches show some promising results in order to cure the PDAC tissue, but as you can see here, compared to the normal pancreatic tissue, PDAC possesses a highly heterogeneous microenvironment. So although SBRT can stimulate some immunogenic tumor cell death, but some tumor cells can mediate DNA damage to escape cell death mechanism. As you can see here we present our experimental setup, and we have both terahertz emitter and detector, which are basically low temperature grown gallium arsenide photoconductive antenna. And we use a Ti-Sapphire femtosecond laser in order to excite the emitter and detector. And we place our sample here in the transmission geometry on a custom holder, which is mounted on a translational stage, XY translational stage, in order to perform a raster scan. If we have a thick tissue, which may be some millimeter thick, and if they are embedded in paraffin, they can be freestanding. But if we have a thinner cross section of tissue, for example, into the micrometer region, it becomes very impossible to make them freestanding, and then we have to mount them onto some sort of substrate. The usual choice for substrate for biological tissue is glass, but we cannot use glass for terahertz time domain spectroscopy because glass heavily attenuates the terahertz wave. So here we tried different sort of substrate and mounted the tissue onto them and tried to look for the optimal one. So here we present the impulse function extraction method. So we have a tissue which is substrate on which is which is mounted on a substrate, and we took a signal using this tissue, and also we take a signal of the just bare substrate without any tissue attached to it, and then we basically align these two signals onto each other, and then we take the first Fourier transform or FFT, and then we divide them. Then we just let it pass through a low pass filter, because as we are doing the data analysis into frequency domain, often we see that there are low frequency noises present in the high frequency domain. So it's necessary to pass it through a low pass filter. Then we took a inverse FFT, and finally what we get is the tissue impulse response. This step is necessary because especially for thin tissue sections, the substrate contribution is very dominant compared to the tissue contribution. So here you can see our effort to find an optimum substrate. So the dry nitrogen trace is basically empty setup when there is nothing in between emitter and the detector. Why we say dry nitrogen is that because we enclose our experimental setup onto our dry nitrogen purge box in order to eliminate any water signature from the atmosphere. And we selected 3 candidates, which is plastic, cellulose acetate, and mica. And as you can see here that the cellulose acetate has like the least time delay compared to dry nitrogen or empty setup, and it also has the lowest attenuation. So we chose cellulose acetate as our best substrate. We also did some experiment with plastic microscope slide because that was our next best choice, and we discarded mica. So as you can see here, the impulse response of untreated PDAC and SBRT treated PDAC, and the first one, number A, is 10 micrometer tissue section on a cellulose acetate. And as you can see, although we can see some difference in the impulse function, but more or less this lies in the noise floor. On figure B, we show the difference between these two groups for a 50 micrometer tissue mounted on a cellulose acetate foam. And number C gives you a 50 micrometer tissue section, which is mounted on a plastic substrate. And you can see here that this B gives us the best distinction between the untreated PDAC and the SBRT treated PDAC. So we choose this combination as the best combination for characterizing untreated versus SBRT treated PDAC samples. And summary of our work and the future direction. So we presented an in-depth study in selecting appropriate material as a substrate to mount ultra-thin biological tissue samples for terahertz time domain experiments, which can be performed in transmission geometry. We additionally established an optimum terahertz tissue thickness in conjunction with the best mounting substrate, which is a 50 micrometer thick tissue sample mounted on a cellulose acetate foam. And we also demonstrated that the terahertz time domain spectroscopy can identify the underlying distinction between the microenvironment of SBRT treated and the untreated PDAC tissues. And our future work will include a high resolution 2-dimensional scans of the SBRT treated and untreated PDAC samples to determine the underlying biology causing the contrast between them. And thank you so much for listening to my talk.